Thank you guys all very much for coming. First, thank you also to Amy, who organized this entire event and is the reason any of us are here and meeting each other. So please take the time to say thank you to her today when you see her later. Like Amy said, my name is Amanda Rickard. I'm a FSHD researcher, and I started here at Genea in February, so I'm fairly new to the team here, although everyone started here at the end of the year last year because our group moved over from Australia. So this space is sort of new to us. Thank you for being patient with being like shoved in the corner in a chair here. We don't normally have this many people in this space, so it's been really nice to have you here. I'm gonna give a talk about what we're doing to develop therapeutics for FSHD here. We're focused on small molecule screening, so those are drugs. And then Annabelle will give you some information about the state of science, sort of, for cell therapy transplantation, which I think was a big interest during our last meeting. So I asked her as a favor to me to come and talk today. So please also thank her as you see her around the lab. After those two talks are finished, we will give you guys a tour of the lab. So Suzanne and Colin are also here representing Genea. Feel free to ask any of us questions. We'll show you all about the different technologies that we're using and try to give you a good idea of what exactly it is that we're doing here. That's the goal. I'm going to try to be as clear as I can about our research, but please ask me questions throughout if you have questions about something specific or something isn't clear or I haven't covered it. It's more obviously really important that you understand what I'm saying, otherwise there's no point for any of us to be here. So I'll talk about disease modeling and therapeutic screening for FSHD. So first, just an introduction to our company generally. So Genea is kind of a global company with 4 different branches. Historically, we're an in vitro fertilization company based in Sydney, Australia. And so that's the blue branch that you see here. So we're a fertility clinic and a world leader in IVF. We also have a consultation branch. Those folks help people set up IVF clinics throughout the world. So we're trying to be a resource there. And then we have biomedics, which designs and sells the different technologies that you would need in order to run an IVF clinic. And then you're sitting now in Genea Biocells, which is our research and development division. So Genea generally has some great resources available to us. We are the world's largest private embryonic stem cell bank. So we have many embryonic stem cells that come from our scientists in Sydney, Australia. Here, we have expertise in neural and muscle development, so we're really interested in taking all of these embryonic stem cells that we have and using them to model diseases using our high throughput and automated screening system. So we harness the power of robots to try and find drug development, to try to work on drug development for muscle diseases and neural diseases as well. So all the different members of our staff, there are about 12 of us, all the different members of our staff have a different sort of project and choose a different disease from our stem cell bank to study. Does that make sense? Perfect. So first of all, why do we care about stem cells? Why do we want a stem cell bank? What are we going to use them for? So the beautiful thing about a stem cell is that it can have 2 different properties. It can self-renew, so we can let these stem cells divide in our dishes as many times as we want to and always get more stem cells out. It can also form all of the different cell types of your body, right? So that's a pretty amazing dual step for the tool that we have. And this is an example of what a stem cell colony looks like, taken by Suzanne, a picture that we took here in the lab, just to give you an idea. Questions about that? So stem cells are a really useful tool for us for disease modeling because, like I said, they have unlimited capacity to divide. We can have as many of them as we need to. That's really important for us when we're plating 100 wells or 300 wells of a plate and trying to put cells in all of them. We need as many cells as we can get in order to be able to test all of the different treatments that we'd like to test. They also don't require a patient biopsy, which is really nice, so we don't have to ask people to go through that procedure, and we don't have to depend on the type of cells that you get out of a biopsy, right? You're only going to get one cell type out of a biopsy, while we can use our stem cells and make them into any type of cell that we'd like to. We can also, because we can form any cell type that we want to, maybe there are multiple cell types that are relevant for a disease. Maybe we're interested, for instance, in FSHD and studying muscle, but also we know that there are changes in the retinal epithelia in the back of the eye. Maybe we could make eye cells from those stem cells as well and study both of them. We can also study the complete development of the cells that we're interested in, right? So we can go all the way from stem cells through all of the different steps that you would need to make an adult cell. And we can study how those steps are affected in each disease that we study. That's really useful to us, especially for certain diseases that have developmental changes. Also, historically, the most successful drug discovery programs have come from cell-based assays like ours that are from human cells, and they're from disease-affected cells that are naturally disease-affected. So you could take animal cells and culture them in the lab and screen those, or you could try to add in or subtract out proteins that are important to make diseases. But all of those are going to be a lot more artificial than just using human cells that already have the disease that you're interested in studying. Does that make sense? And lastly, just for this specific disease, FSHD the gene for it doesn't exist outside of primates. So it's hard to make a mouse model of FSHD for that reason. You have to add in that gene in order to make it work. So here we can naturally have the disease phenotype in our cells. So the disease is actually there in the dish without us having to add anything. So we were contracted by FSHD Global, which is an advocacy organization based in Australia, to figure out a way to make our stem cells, which you see there at day 0 on the left, into mature muscle cells in order to try and see if we could study this muscle development process in FSHD. So we generated this platform. It takes 26 days for us to turn a stem cell into a muscle cell, going through each of these different stages that are important in development. Questions about that? Yeah, so this is a normal cell. Yeah, so we developed the platform in our normal cell lines, and then we applied them to our FSHD cell lines to see if we could model the disease in a dish. So some background about FSHD. Like Amy said, there are 2 types, 3 types, 4 types, probably more than that, to be honest with you. But the place where they converge is that the different mutations for these different diseases result in expression and turning on of this DUX4 protein. So all of them kind of converge there. And DUX4 protein sets off a slew of different problems in the muscle cells in which it's turned on in FSHD patients. We're really lucky because many of the steps of this slide weren't even published when I first started studying FSHD. And so we've come a long way in the last few years. And the nice thing about it is it gives us many opportunities for intervention. So our group, like I said, developed this human embryonic stem cell model of FSHD, which we published earlier this year in Stem Cells Translational Medicine. An important point, obviously, was that DUX4 was expressed in our FSHD cells, and we couldn't find it in our control cells, which is exactly what we would expect. And that's what we need in order to be able to track the changes that DUX4 is making to those muscle cells.
Did you get tissue then from an FSHD patient?
So these are actually embryonic stem cells that are differentiated to muscle.
How did you get DUX4 to express that?
DUX4 normally comes on in an FSHD patient and during this stage from the transition to myoblasts to myotubes. So when we put our FSHD cells through this process, then we can see DUX4 in the myotubes here.
So that was your question. Did they have FSHD tissue cells?
Not tissue, starting at stem cells.
So the stem cell is coming from a FSH patient?
The stem cell is coming from our in vitro fertilization clinics in Australia.
Why is DUX4 on then?
Because the cells have FSHD. So if you make muscle from a cell that has FSHD, you'll see DUX4 expression.
But if you start out with a normal, I thought you said you start with a normal patient.
We developed the platform in normal cells,
Oh,
but then we've applied it to our FSHD cells to try to model them. Yeah, good question. Okay, so we could see DUX4 in only our FSHD cells, but not in non-affected controls, which is exactly what we would expect. We also found some muscle cell defects, which we published in this paper. I'm not going to go through all of the details of the paper, but just so that you understand a little bit about what we're doing. We found, that wasn't surprising. So we found muscle cell defects. So the cells in this top graph behave differently from the way that we would expect them to if they were unaffected. And they also had a different size and shape than the ones that we were comparing them to in unaffected. Then what we did was looked through all of the genome to figure out which genes were expressed in the 2 different types, the unaffected or the FSHD cells, to see if we could find a difference there. And each of these bars represents a gene that was different in FSHD compared to controls. Does that make sense? Green means they were up and red means that they were down. Meanwhile, so that was happening in Australia, and Dr. Leslie Caron was lead on that project here. Meanwhile, I was in Seattle, and I was developing a tool to measure and track DUX4 in live cells. So I was at the University of Washington in Dan Miller's lab. We used primary patient samples, so that came directly from a muscle biopsy. And what we did was develop this viral tool that you can see the schematic up here, where if DUX4 was present in an FSHD cell, that cell would turn on either blue or green were the two options for us. So you can see that here. These are muscle cells here in green. The red dots are the nuclei that have DUX4 in them. So that's the DUX4 protein that we're staining. And then here's my tool being expressed, showing you that those cells are also blue, which is really nice for us. So before we would have to fix the cells and kill the cells essentially in order to be able to find DUX4 there. Here we could look in our microscope and know exactly which cells were expressing DUX4 because they were glowing.
Now, I'm going to ask you a question here because this whole
Please.
thing with the genome thing and the DUX4, I gather the generation or the occurrence, say, of the DUX4 issue is the bad part. Is that what makes
Yeah.
it go active and the symptoms start?
Yeah, that's exactly what we think.
Amanda, would you repeat the question so we can get on tape?
Sure. So she was asking if DUX4 is the bad part, is what's happening to the muscle cells that's giving people problems. So we think so. So researchers have shown that the DUX4 protein alone causes a list of different problems in cells, and people are still reporting these, right? That's what the et cetera is for. There are probably more than this list. So it does things like activate cell death mechanisms and change the way that the cell looks generally, just like I showed you in those red and green graphs. So a lot of changes are happening to FSHD cells when DUX4 is there. Yeah.
So DUX4 is bad.
Yeah, exactly. I should have a shirt that says that. Please.
The one on the right is obviously FSHD.
Yes.
You turned on so they indicate through color, so you can see them.
Yeah.
On the left, what are we looking at?
So the left is our
Those also DUX4 cells.
Yeah, good question. So the left is our unaffected cell line that we're running next door. So our healthy cell line is what we would refer to it as. And then the right is the FSHD cells, which automatically turn on DUX4 when you make myotubes like this with them.
See it until you.
So we could only see it if we applied this stain, but not in live cells. So we wanted to be able to go into live cells and watch them under our scope and see exactly what's happening to them when they express DUX4. And so what we ended up being able to do is follow these cells during the time of muscle differentiation, and we could track when they were turning on DUX4, how much DUX4 that they were turning on, and we could graph it here, which is this dotted line. And then we could also see that, and I'll show you a video in a little bit, we could see that when cells turned on DUX4, about 20 hours later they would die in our tissue culture dishes, just the cells that were DUX4 positive, which you can see quantified here in the black bar. So now we felt like we had a really nice model of FSHD happening in our dishes that we could move on to drug screening. So when I came to Genea in February, I brought that reporter with me and looked through all of the different cell lines that Genea has here for DUX4 expression using the reporter that allows me to visualize the cells. So we have 3 different FSHD lines. So my first job was to optimize our FSHD model and make sure I could find as much DUX4 as possible, and I could detect it as well as I would expect to be able to. So all of these different graphs are sort of different culture conditions that we tried growing our muscle cells in, and different cell lines that we had here in the lab that we were testing to see which of the conditions gave the best DUX4 expression. So we're trying to make the best possible model so that then we can use it to see if we can knock down DUX4. Does that make sense?
When you say the best DUX4 expression, do you mean that you have more of the DUX4 protein present in that cell?
Yeah, exactly. Well, we mean that we have more of the cells in our dish that are expressing DUX4. So the way that you treat the cells will influence how they express different genes. We wanted to treat them the way that would allow us to see the most amount of DUX4. Yeah? Okay. So lots of different conditions that we tested. And what happened in our cells was that as they differentiated over time, which you can see here, they eventually turned on DUX4, and we could watch those cells die here as well. So I'll show you a video, and we'll eventually see the green cells pop up, and we'll bounce around a couple areas of the well so you can see how many DUX4 positive cells there are. So this was really exciting for me because now I could track, right, which of our cell lines was the best and which of our treatments was knocking down DUX4 expression. So this is during that very last phase of muscle differentiation. So this takes about 4 or 5 days to start to see the differentiated mature muscle. Yes, ma'am. Yeah, I've got a quiet voice naturally, sorry. Okay, so now I could track DUX4 expression in our cells here at Genea. I could make as many of those cells as I wanted to because I'm starting with stem cells, and I could move forward to try to figure out what sort of treatments we should apply to these cells to see if they were going to work. So this is the same schematic before, only now I'm showing you sort of all of the different ways that we're trying to attack this progression in FSHD, right? We can start with the DNA and try to influence it with the drugs that we're using to stop it from expressing DUX4. We can target DUX4 directly, right, and try to stop it from doing anything downstream. And then we can also target all of these different things that DUX4 is doing to see if we can improve the cells in our culture. So now, so it took quite a while, but our chemists worked hard to develop and collect a whole huge library of compounds, which we will show you when we give you the tour of the lab, that we'll put on our FSHD cells and see if we can stop them from turning green.
And the expression of DUX4, just make sure I understand, this
Please.
is endogenous expression.
Yeah, exactly. So endogenous means that it is naturally coming from a place where you would expect it to come in the disease. We haven't added it or done anything unusual to make the cells just naturally express it.
Do you have a sense of the levels compared to other proteins?
So it's sort of DUX4 itself is a rare protein. And so it's certainly not on in 100% of our cells. In fact, maybe 1% of our cells will usually express DUX4, somewhere around that level. It depends on the different cell lines, and it depends on the culture conditions that we treat them with. So how do we grow the cells and all of that? Does that make sense? Yeah.
Can you be reasonably confident if you found something that would knock down the DUX4 that it would behave that way in an adult muscle cell?
It's an excellent question. So we're trying to find ways to prove to ourselves that any treatment that we find will be relevant once you get into an adult. And the way that we do that is by defining a whole bunch of different measurements that we can do when we put the treatment onto our cells. So we can measure DUX4 protein directly and see if we're allowing the protein to knock down, which you can see here pointed to with the arrows in muscle nuclei. We can look for all of the different genes that we know that DUX4 is turning on in the genome and see if those ones are brought back to that unaffected control level, right? We can stain for those. We can also do this same RNA sequencing platform that I was showing you before. So hopefully any treatment that we give would make these cells look more like these cells over here, right? And we can assay all of those genes and ask that question. We can also do live cell imaging, watch through our scope and see if we improve the muscle cell death. So maybe we don't knock down DUX4 directly, but instead we just stop the cells from dying. We'd be pretty happy with that. So we can measure that as well. We can measure whether or not our muscle cell size and shape in our dish is improving. And then we're also collaborating with researchers at the University of Massachusetts in Charlie Emerson's lab to develop a small animal model of FSHD. He's handling that for us, so we'll be able to move directly into a model that's got FSHD DNA in it. So instead of adding the DUX4 protein to the mouse model ourselves, we're instead engrafting human muscle in, and then we will add the treatment there and see how it improves the mouse. So we're hoping that by combining all of these different things together in our drug screening, that we'll be able to make sure that therapy is doing exactly what we expect it to be doing in all of the different cell types that we tested in. Yeah. So just a quick overview. Our goal was to develop a disease in a dish model of FSHD, which we've done. We wanted to optimize cell conditions so that we could detect DUX4 the best, which we did using that green or blue tool. We wanted to define measurable outcomes. So those are all of the different things in my previous slide that would allow us to actually measure whether a treatment is making a difference for FSHD. And we wanted to design and compile drug libraries that were targeted very specifically for all the different pathways that we think are involved. So that's important because there are millions of drugs that we could test in the world, right? So it's much better for our time and for our money to specifically design and choose drugs that we think will have an effect, which we've done. And so now we're moving into screening compounds for FSHD. So hopefully the next time that you guys come to visit us, I can show you some of the data that we've done so far. I wanted to mention that a lot of this work was funded by Friends of FSH Research in Seattle, and the original development of the muscle differentiation and from stem cells was funded by FSHD Global, so we really appreciate both of their support. The RNA sequencing, genome-wide RNA sequencing was done by Dr. Lee at the National University of Singapore. David Gabellini did the DUX4 measurement in our original model of FSHD, and the tool that we're using to light up the DUX4 positive cells came from Dan Miller at the University of Washington, which is also where I came from. This is our team of incredibly talented people that I feel very lucky to work with. I'll just point out the folks who are specifically working on FSHD with my pointer here. So second from the left is Alex. He's our chemist, so he designs all of the drug libraries for us and has a pretty impressive background in drug development for diseases. Uli is our fearless leader here. Let me see if I can, this guy. He is the boss of everyone that you see in the picture, and the reason that, that we're here today in San Diego screening for FSHD, certainly. Annabelle is here in the front next to Uli. She'll be speaking next, and she's also on our FSHD drug development team. And you guys know me. And Charles is also working with us because he's all the way to the right here. He's our automation specialist, so he keeps all of the robots aligned and in order. And lastly, the newest member of our team is working on FSHD is Monica, and she's our preclinical development director. So she helps us move from the lab into the clinic. Yeah? Does that make sense? Questions?
So, everybody, excuse me, that has FSH has the DUX4?
So yeah, so far, anytime that we look into FSHD muscle, if we culture that muscle in a dish, then we find DUX4 protein there at some level. But it's really variable, right? Just like the disease is variable. Right. So some folks who donate tissue, we see a lot more DUX4 expression, and some we see less.
Okay.
It's sort of,
you see that with FSH2 and as well? You see the DUX4?
Yeah, we see DUX4 expression in FSH2 as well. In fact, sometimes more DUX4 expression because sometimes it can be more severe depending on the person.
Is there any correlation between clinical finding and DUX4?
What do you mean by that?
Well, the more severity in the disease, does it show also in DUX4?
Yeah, so the question is, if there's more severity in the disease, will the person who donated the biopsy, or will the muscle that we grow in the lab have more DUX4? That's what you're asking, right? DUX4. So it's kind of a hard question for me to answer because all of the samples are blinded by the time they come to me to measure DUX4 expression intentionally, right? I don't know that I have any one specific sample, but generally we do find more DUX4 expression with shorter arrays, which also correlates with, so that's just the length of your piece of DNA, which we can talk more about later if you're curious about that from diagnosis. But shorter arrays usually mean DUX4 expression, and they also in the clinic usually mean more severity, and so we can kind of correlate them that way. You have a question?
So early on in my diagnosis of FSH, I was going and still go to Dr. Kaplan in Encinitas, but I started noticing elevated levels of CPK. Can you tell me what the relationship is between that and DUX4?
Yeah, so he was asking what the levels of the CK levels in the blood have to do with DUX4 and why they would be elevated in FSHD patients. So we see that consistently, elevated CK levels, and we see also that it correlates with disease severity. So the more severe you are, the higher your CK levels in your blood are. And that's common to most muscular dystrophies. So CK is a measure of whether or not a muscle is breaking down and turning over. And so we would expect to see that in FSHD, but it's not a specific marker of the disease, if that makes sense.
So the protein, the DUX4, is actually causing the destruction, the CK levels.
Yeah, we think so.
That the destruction has occurred.
Exactly. Yeah, exactly.
Also, you may see higher GGTP, but people think it is a liver problem, but has nothing to do with the liver. Among the physicians, they think it's a liver. They don't have enough knowledge that it may be from the muscle. GGTP.
GGTP might be from the muscle instead of from the liver if it's elevated. Okay, so that might be another one to think about running for a blood panel. And then I can show anyone who's interested in this, but we published a paper in the lab at the University of Washington where we went through and assayed a couple thousand proteins in the blood to see if we could track any that increase with disease severity. CK was one of the ones that we identified, which is why I know that, but there's also a list of other ones that are there. And so those will be really useful for us once we moved into trials, because we can track whether or not a disease is getting worse or whether it's getting better using the blood levels instead of having to go into the muscle, which would be much better, obviously. Yeah?
So if you develop a drug and it's going to target the DUX4,
Mm-hmm.
that would prevent further damage, but it's not going to do anything for muscle death already.
Yeah, so Annabelle will talk a bit about muscle stem cells, but muscles have stem cells inside of them that are meant to repopulate muscle when it's damaged. And so muscle's really good at rebuilding itself because its job is to be a little bit destroyed each time that you use it and then rebuild stronger. So we think that those stem cells that are already in the muscle, as long as DUX4 isn't being expressed there, will start to fill in as well. And then Annabelle's going to talk about potentials for cell therapy, which would be transplantation of muscle cells to replace ones that are lost, and how that's going in different muscular dystrophies. So those would be, kind of the two ideas. Cool.
Amanda, talk a little bit about FSH Connect.
Champions? Yeah. So the FSHD Champions is a really exciting initiative, I think, where a lot of the different FSHD advocacy organizations around the world are coming together and realizing there's probably strength in numbers here. We don't need to be repeating all of the things that we're doing in all of these different countries. Instead, we could have sort of a global platform for raising money for FSHD, for funding science, and also for putting together a global registry for patients. So that's a really important focus of their time right now. It's important because FSHD is relatively rare, so if we want patients for clinical trials, we're going to have to be ready to contact them as soon as we're ready to move into trials, and we can't do that right now because we don't have a global way to get in contact with people. We also don't have a global way to track the changes that people are having in their own disease progression, which would be really useful for us to know, right, how it actually progresses in each person. And it will give us more opportunity to break people into different groups once we have more information on them so that we can specifically target therapies to those groups that might benefit most from them. So that is one of the initiatives of the FSHD Champions, but overall it's an effort to bring the different organizations together for hopefully better benefit for everyone. Yeah. Please. What do you mean by the other diseases?
More like, actually more than your research on us. Like, we are getting old, we got other things, other, effects on our body, the pain, back surgeries,
Yeah.
this and that. How all that affects?
I don't really have a good answer. Oh, sorry. He's asking how would other diseases or conditions affect FSHD specifically. So I don't really have a good answer for that because I'm not a clinician, but overall we know that things like damage to your muscles, like C-sections and sort of trauma and that kind of stuff, is going to make them harder for them to regenerate and repopulate, especially if DUX4 expression is there. And we know that the disease gets worse over time, right? And so you would expect to see worsening symptoms the older that you get. Otherwise, there hasn't been a lot of research on what we call comorbidity, so two different diseases in the same person, and how that would affect FSHD progression. But it's a good question, and I think now that we've got more researchers and more funding, we'll start to answer it.
Well, FSHD itself brings other diseases like arthritis.
Sure, yeah, exactly. People have complications beyond just muscle weakness, yeah.
So in this case, do you see the difference with a person who has FSH and arthritis and a single person with FSH?
Sorry, I didn't hear the first part.
Yeah, like that is a good example. Like, do you see any difference between a person who has FSH and arthritis and a person who has only FSH?
Yeah, I don't specifically see a difference because I'm not allowed to know if someone has arthritis, but and I haven't seen any papers on it, so I think it's something that, yeah, should definitely be addressed, but we don't really have a good answer right now. Good question.
That could blow your mind if you start throwing in all that extraneous issues that
It's complicated
everybody will
enough.
have.
No, it's
Yeah,
important though.
gosh.
There are a lot of
Microfocuses.
efforts. There are a lot of efforts in the clinic
Yeah.
just to identify properly a patient with FSHD and to really classify type 1 or type 2 because it's already having so many symptoms as all muscular dystrophy that basically is misclassified and mistreated usually. So still there are efforts just to really classify what are the symptoms,
Yeah.
the key symptoms, and what are the key markers in blood or in other maybe muscle biopsies, just to be sure what is the diagnosis. And maybe clarifying all that it will come out if it's linked to another problems, like arthritis or maybe another symptoms.
Yeah.
Talk to me.
We
Jen.
don't know
Talk
the
to me.
we don't know the answers yet.
Amanda, can she repeat that with the mic?
Sure, yeah, I'll give her back the mic. So let me introduce, this is Dr. Annabelle De La Garza. She's our resident MD PhD, and she came from the Children's Hospital in Oakland, where she studied muscle stem cells. So when folks mentioned that they were interested in that idea, I thought that's not my specialty, but certainly Annabelle will be able to give us a talk on that. So please welcome her warmly, and I ask questions of her as well.
Well, good morning, almost, afternoon already. It's really a great pleasure to have you here over to show you what we are doing. I think it's of great interest of you what is behind the scenes of research. So as Amanda mentioned, I'm coming from the Bay Area, from Children's Hospital, that I was just focused on muscular dystrophies, Duchenne, with cell therapy in animal models, basically. And before that, I was in Holland, in the Netherlands, in Leiden University, where I did my PhD, focused on stem cells and also muscular dystrophies. So, well, here I was recruited last year. Now, as Amanda previously mentioned, it's very a short time that we are here, Genea. They just moved from Australia to here, and it's where we all just start gathering here and start our work with muscle, and to be able to move this forward, possible to translate it to the clinic. So here I just want to mention like an overview what is going on with the cell transplantation for muscular dystrophies. Please feel free to interrupt me whatever time if you have questions. So just to start this presentation is, well, to give the introduction of our most abundant human tissue, which is the skeletal muscle. As you know, we have muscle all over the body, and there are different sizes, and most of them that are very accessible, that make it very good candidate for protocols that can be for the delivery of stem cells or any other compounds or treatment. To note here in this diagram, there is not depicted the diaphragm or some intercostal muscles. The diaphragm is one of the muscles that really helps you to breathe, and that is a little bit tricky and challenging to approach. But as you see, all muscles are very easy to access. Muscle disorders, we can group them in many different classifications, depending on the cause, the origin, the development. But just to keep in simple words, we can say that it's acute and a chronic disease. So the acute disease is basically related to trauma, related to tissue damage or loss. This can be just as simple as a hike that is 3 hours hike, 8 hours hike, you can already feel the pain, and that is already a damage in the muscle. But that is normal, and we have stem cells on the muscle, and they will just overcome with that pain. Other acute examples can be like some fractures or some car accidents where you have more compromised, bigger areas of muscle. But in general, we can say that they resolve easy and okay. In the chronic condition, here for the acute, what is happening in a very simplistic way is our key players that are stem cells of the muscle called satellite cells. Those guys are dormant usually. As soon as we're born, they are just in their place in the muscle fibers, and they are just dormant. You have a trauma, as I said, an exercise or an accident, whatever, and these cells, they immediately get the cues to get activated, which means they proliferate, they migrate to the area where is the damage, and they join to patch these damaged areas, this damaged muscle, or they can make the newly muscle fibers. So this cycle is normal, and it can be as soon as like maybe 30 days or so. But what happened in the chronic condition, that here, one of the examples is the muscular dystrophies, that most of them, they have a genetic background influence, like Duchenne muscular dystrophy, limb-girdle muscle dystrophy, and FSHD, facioscapulohumeral dystrophy. What is happening here is that this cycle of regeneration, the activation of the satellite cells, is continuously, because you always have the damage in the muscle. It's not cured because the cause is more severe, no, it's genetic. And it's well described in Duchenne that these satellite cells, they reach the exhaustion period and they cannot really make more muscle. And what is happening is that this tissue degenerates and creates fibrosis, which is just connective tissue, fat, and then makes it even harder to cure this disease. As general terms, what we can talk about the treatment for these chronic disorders is basically the conventional treatment. It is very supportive. And physical therapy is one of the first indications that are given to the patients. Some orthopedic appliances that they might need it according to the severity and the progression of the disease, even wheelchairs. And also, as I mentioned, there is an inflammatory component that also in the chronic condition, because it's happening this constant damage. So there are a lot of infiltrating cells, inflammatory cells, macrophages, leukocytes, and so on, and they even make worse the condition. So they really make it a big disaster. So the steroids is one of the drugs that is so far the best just to reduce this immune response. And of course, there is a lot of room for ongoing research to really approach or get a more curative treatment. And for this, there are the small molecules, RNA therapies, and gene therapy, that basically that might be the most curative treatment in these muscular dystrophies where the genetic component is involved. And cell therapy, that in combination with gene therapy, I think that is the most promising approach. So for the talk of today, I just want to mention the cell therapy. So to give you a little bit of historical aspects, what happened even in the early 90s. So one of the things was to choose what would be the best cell to transplant and to make muscle, to repair it. So the satellite cells, the stem cells of the muscle, that also myoblasts and is the progeny, were the ones that were selected. And these ones, they went through the clinical trials, and the idea was to collect a muscle biopsy from a healthy person, just isolate the satellite cells and implant it into the muscle. How? Just injected it intramuscular and hoping in a few days that these cells, they will integrate into the muscle fiber, and you can see it like a patching or integrated into the muscle. No, as you can see it here, a little bit brownish. So it was a great approach. It is a great approach. But at that time, there were many issues that were not really fully convincing, especially because the outcome was the limited gain of the muscle strength. And most importantly, is for the cells. They didn't survive long enough. They died very quick, and they didn't migrate. They basically stayed where were injected, and there was rejection, the immune rejection. But the biggest problem of this is that the cell availability. As I said, the muscles, the cells that were taken were from biopsy. And a biopsy, well, it's small, no? You cannot ask kilos and kilos from donors. And so in the coming years and nowadays, there are some improvements in this transplantation that involves immunosuppression, just allow these cells to stay longer, and also the high density injections, which means that instead of where at that time they were 5 millimeters separate, the injections now is 1 to 2 millimeters. They are closer, and also to give the cells in less volume instead of large volume, because that also the volume, it can make even more damage into the muscle. So there have been some improvements in the entire therapy. But however, until now, we can really consider that this immune match transplantation is only for localized forms of muscle disease, like is in the case the oculopharyngeal muscular dystrophy. This disease is affected in patients with affected the pharyngeal muscle that basically cause them pain when they eat, and also ptosis. And ptosis is that the lid of one of the eyes is just all the time down, so they cannot open it. So this treatment is great because there are small muscles, localized, and works perfectly. There's still just in phase 1 and 2, just safety and efficacy, but it will move forward to phase 3. Just to briefly mention, I'm not going in details unless you might have questions, then we can discuss later on. But there are a bunch of different cells in order to approach the problems that the satellite cells were having as a cell transplantation, like the migration, the expansion of the cells, and taking the source of the cells. But the major problem still is the availability. If even all these cell types, still you take very little amount, and then you need to expand them in culture that is not the best, and then be able to transplant into the patient. So one of the measures, as I'm considering here, is the availability of an adequate number of stem cells. And also you need to have large scale amplification in culture, especially for all these genetic disease that is not only one muscle affected, there are many. So you need a lot of cells. And other characteristics that are important to consider if you want to select one cell type is that these cells, they can really make muscle, and it means the myogenic potential that is possible to modify genetically if it's needed for depending which gene is mutated and for each disease. Also to choose the administration route, because I just mentioned the intramuscular injections, but if you want to reach many muscles, especially for the diaphragm, you have to go for systemic delivery. And also the immune response, and that is very important to consider, that it might be different from patient to patient. So it's something that has to be analyzed and detected individually per disease and per patient. So to overcome with all this, what we are working and that we are saying, and is a new approach of pluripotent stem cells, is, well, first of all, what is a pluripotent stem cell? It's a cell that basically can make all kind of cell types in all human body. And on top of that, it has the unlimited expansion. So for all these reasons, it can be really a potential source for regenerative medicine and tissue replacement after injury or whatever disease. So it means that these pluripotent stem cells can make not only muscle, but if we want only, we can make it to muscle, but also we can cover other cell types that sometimes are needed when you are doing a cell transplantation. A certain degree of the disease, sometimes the muscle is not the only affected. There are more cell types that are involved, like blood vessels, for instance. So from where are they coming, these pluripotent stem cells? We have two major sources. One of them, that is what here at Genea we're working on, is from the discarded embryos that are coming from in vitro fertilization procedures. And of course, with informed consent of the parents, we are taking these embryos, and they are like 3 to 5 days. We take just the inner cell part of this embryo, we put it in a dish, we just expand them, and it's what we call embryonic stem cells. And those ones are our super great star. Most recently, we have another way that we can create these pluripotent stem cells. It can be as simple as just from a skin biopsy. And with this little small piece, just you dissect and isolate, just like a fibroblast in this case. You just expand them, and you mildly modify using at least three reprogramming factors, and then you make them more embryonic conditions. So they are coming from an adult, and then you are converting it in more embryonic cells. And those ones are called induced pluripotent stem cells. These technology is very recent discovery. It was in 2007. And Dr. Shinjo Yamanaka, who is the one that discovered the factors on the use of this technology, he won actually the Nobel Prize in 2012. Well, but here at Genea, we have these pluripotent stem cells, and you already seen this diagram that Amanda showed you. So we have the embryonic stem cells that we can really, with the protocol, the platform that we have developed to make muscle, we have the entire development of the muscle, so we can pick and select which cells during this development we can study further, not only in healthy conditions, but also in disease conditions. And ultimately, we can select these cells and just bring them to a specific disease and being possible to translate it towards the clinic. Of course, to jump from the cells and into the patient, there are many steps to be considered. And that is what most of our efforts are for now, just to study more detail, like what is happening with FSHD, in order to be able to select which cell type would be the best to approach a different disease. So in conclusion, we have that the cell transplantation for muscle disease is possible. It's really needed to select the right cell type and to have large quantity of it. And the pluripotent stem cells are the great unlimited source for different myogenic cell types. So, if you have any questions?
Oh, that's cute.
Did you mention that you use a steroid for therapy?
The question is if if we use steroids for treatment. We do not treat with patients. We do not deal with patients at this stage. So that I cannot give you an answer. Yeah. But our cells, our cultures, we do not put any anti-inflammatory or any of those drugs when we are working in culture. Mm. Mm. Yeah.
Could you summarize for the microphone?
Yeah. So the question was, how long will it take to go for these compounds towards the clinic? One of the answers, is that it all depends how is, of course, the funding and the progress on research, but also depends on all the knowledge that we can get about the disease to be able to apply it, and also the compounds that we have. Some of them, they've been already used in other conditions, so still it's like it's safe and approved in certain conditions, but doesn't mean that it will work in the other condition. So still it's a long process just to get into that. And here we are still working with collaborators in order to go for more preclinical studies, no? Which means like the animal models that we need to be sure on confirm that disease can be present on there. And further on we can go for clinical trials. But it's still a long way to say. A specific time is hard to say, but we are on their way.